Today I'm introducing a new concept called the Hardy-Weinberg principle. This principle is essentially a way for us to look inside a population and look at something called microevolution. See, we've been studying the larger effects of evolution over a slow amount of time. We know there's modifications as we descend from one species to another. There's descent with modification. And this is normally very slow because it takes a what? A mutation, which we've learned is a very slow process. And then that mutation has to be advantageous to a changing what? A changing environmental stress that makes that allele, that new piece of genetic code, okay, come to the forefront and increase its frequency as the others in the population who do not have that allele cannot survive with the new changing environments. That's important that we understand. So this principle is a way for us to look at the micro changes. We've been studying the macro changes, for instance, the changing of the skulls in the hominids, or we looked at having a stickleback spine or not. Those are big changes. But we're going to talk about the micro changes in an environment. People have asked me, are we evolving right now? And the answer is definitely, but not in a large macro scale. Because we're looking at a small section of time, the changes we're talking about are micro changes. So we're going to look at the frequency of alleles. Now, we talk about alleles, I can't get away from the fact that we're talking about chromosomes. And we look at the fact that most of our complex organisms have a diploid number, a pair of chromosomes, where we could have, let's say, a locus here, we have an A, okay, loci I should say, and let's say a recessive gene B, so a dominant A and a recessive. Now I know there's more complex genetics we've talked about in terms of codominance, okay, or incomplete dominance. But you know, it always comes back to what? It always comes back to the fact that we have a pair of chromosomes and that you can either have one or the other gene. Now, certainly genes do work together. We've talked about the regulatory genes, but it always comes down to what? It comes down to the fact that we've got a pair and we either certainly have one or the other type, let's say, of gene or chromosome or allele, we should say, which is the expression of that gene. So even though you may think, gosh, this is the simplest form of genetics, it still rings true in many of our disorders. Case in point, we're talking about PKU today. Now, PKU, as I talk about, is a rare inherited recessive disorder. And there's a bunch of them. Hemophilia or sickle cell anemia, where the red blood cells are in a sickle shape. And those are among many that are recessive disorders, meaning what? Meaning in order to have the disorder, we have to be homozygous recessive, meaning we have two broken genes. Remember, the recessive is normally a gene that's broken somehow, that doesn't work as well, and A is the working one. So although this is the simplest form of genetics, it still rings true in a lot what we talk about. And a lot of our genes are either one form or another because of this diploid number. So this works very well. So as I talk about today, about PKU being a recessive order, and let's talk a little bit about it because it's important you realize that these genes on this chromosome at a certain spot, loci, okay, code for amino acids that build proteins. So in any case, this rare inherited recessive disorder causes an amino acid called phenylalanine to build up in your body. And of course, that becomes a very dangerous scenario. PKU is caused by a defect in the gene. It helps create an enzyme. That's important. Enzyme is actually a protein, and a bunch of amino acids that this gene codes for makes a protein. So as we talked about, it's important we understand that these genes, okay, code for proteins that often or not are enzymes that help chemical reactions occur in our body that we need. In this case, if you are B and B, if you have the recessive two of the what? Genes for the broken gene, you can't make the correct enzyme to break down phenylalanine, and therefore it builds up. And this dangerous buildup can be deadly. okay, if they don't control their diet. So again, this is an example of a disorder that is recessive, and there's many of them. So we can actually track the frequency of the allele of the little b in the entire population with a little bit of mathematics and understanding we're dealing with Hardy-Weinberg principle. Now, before we start about this microevolution, it's really important that we talk about a baseline. Now, to know if in fact a population is undergoing evolution. And again, we're not going to see in a current population over a 10, 20, 30, 100, 200 year change. We're not really going to see much, if any, of macroevolution. I know we've had examples where we have in terms of the finches, okay, or let's say the moths, the peppered moths. But in this case, what we're looking at is micro changes. And the micro changes we're going to be looking for is the frequency or the percentage of the individuals that have this, or yet very important, the frequency of this allele total. All right. So in order to have a baseline, in order we can understand that there's a change in the amount of BB. Now, again, I'll explain. If you have individuals that are homozygous recessive, BB, you can have individuals that are carriers, right? They're heterozygous. And then you can have individuals who are dominant. Okay, these are all different individuals. But what we're looking at in this micro changes, we want to count how many of these total alleles exist. What we're really doing is counting the total number of what? Chromosomes in the population of individuals that are carrying, in this case, the recessive or broken gene. Now, if we can see if they're changing, we need a baseline to know what that is. So the Hardy-Weinberg principle states we're going to assume that there is no equilibrium, there is no change in these frequencies. There's no selective pressure in this case. And what does that mean? Well, in order to be able to find this baseline, what the current value is, if there is no evolution, understand that. We have to know what it is before there is any evolution. That's the baseline. We have no evolution going on. So understanding that, there are 5 scenarios or prerequisites we have to have in order to use this type of mathematics that I'm going to show you. Number 1, and very simply, in order to have a baseline and measure what the actual percentages of, let's say, the P chromosome in a population is, I can't have it changing. So there can't be any mutations. And you know, mutations are very slow, so if we're looking over a population of a couple of years, the chances of having mutation is very, 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 very small. Okay, as we talked about. We're going to say that there's random mating. What does that mean? Well, there's no selective mating. People are not basically mating or finding mates or wives or husbands based on the idea that they have PKU or not. You're not saying, hey, do you have PKU? If not, oof, I can't. So there's the random mating is going on definitely in our populations. And 3, no natural selection. And you'll say, well, what do you mean by natural selection? Well, because we are understanding people that have PKU and we're helping them follow a diet that limits that amino acid that can be deadly, people aren't dying of PKU. Therefore, we're not lowering that number of people who are homozygous recessive because they're dying of that disadvantaged type of illness. We're helping most of them. So there's no natural selection. No extreme, we have very extremely large population. This is important. We're going to talk about something called genetic drift soon, but generally speaking, the larger the population, if a couple of people were to die naturally, it won't affect the percentages in the entire population. When you have a small population of a few individuals and just by chance a couple of them die, that could change the percentage of the alleles dramatically. Think about 3 individuals in your population. What if these 2 were to die? Then you lose all your what? Recessive. So the larger the population, the less chance you have of having a change in the frequencies by chance events. And no gene flow, meaning we don't have a tremendous amount of new alleles entering. So these populations are generally isolated. Okay, and although we do have migration of people in and out of our society, we could suggest that these individuals aren't really adding any new alleles. So these are the conditions that we have to be met in order to come up with this baseline. Again, what's this baseline? This baseline is an idea that the frequencies of these what? Of these alleles, or we should say, again, don't lose sight of these genes on a chromosome. So what's the frequency of having this recessive gene? What's the frequency of having a dominant gene? That's what I'm often about. And then we're going to ask about the frequency of the individuals, and that's a really important. But as we go forward here, understand it's about the frequency at this point of the chromosomes that have certain genes. That's important. And again, if we follow these five requirements, they will be steady. And from that steady number, we get a baseline. And then we can compare from this steady amount if in fact we have microevolution, if in fact they are changing. Okay, now in terms of pKa, like I said, we can assume that the five requirements for the Hardy-Weinberg principle or equilibrium, we'll make more sense of that word equilibrium later, is that the pK gene mutation is very low. So therefore, there are no mutations. Mate selection is random with respect to whether or not an individual is a carrier. We don't ask our potential mates, hey, do you have PKU? We don't ask our potential mates, basically in our society, hey, do you have good vision? All those things. So there's no real there's no selective mating going on. Natural selection only occurs in a rare homozygous individual who are not following dietary restrictions or not following the doctor's orders. Okay, population is large, we explained that, and migration really has no effect because most other populations, most other humans have similar allele frequencies. So we can make that assumption here. Okay, now it gets a little bit tricky for some people. So what do we do with the math here? Well, as we've talked about, and as I will continue to talk about, when we're dealing with all the possible combinations of all of the alleles, let's put it this way, and we'll make it A, big A, and little b, okay, we know that all of the alleles, all of the chromosomes that have the dominant and all of the chromosomes that have the recessive, if you add them up, okay, you'll get a total amount. Now, if we're going to use probabilities, we would take the percentage, or we take the fraction. Let's say half of all the individuals have the dominant gene in one of their chromosomes, and then we would say that all of the chromosomes in the entire population would also have to be 0.5 to equal a total of 1, right? So this is essentially the addition rule, where we have mutual exclusiveness, that we're dealing with totals here. So if I didn't say this clear enough, all of the dominant genes plus all of the recessive genes, and if you want to think of it this way, all of the chromosomes that have the dominant and all the chromosomes that have the recessive, if you add them all up, you get some total. And if in fact 50%, and I'm just making a number up to make some sense here, of all the chromosomes have the dominant gene, and obviously then you have to be 50% would have the recessive chromosome. It's how it works. And you would add those percentages. 0.5 is 50%, 0.5 is the fraction. So when we're dealing with probabilities, we know that we have to always equal 1. Have to, because these are the total choices. We either are going to have the dominant or the recessive at this loci in some chromosome. And that's what Hardy-Weinberg is talking about. He's talking about a single allele at a single loci and a chromosome. And it's either one or the other. Why is it one or the other? Because we have pairs of chromosomes. And that's why it works. Now, so we're going to use the addition rule. And it's very important that you understand why we use it. Okay, so the addition rule works, and we add those probabilities because it's a total. Those are the total choices, total amounts. If I say to you, I throw a 6-sided die, what's the chances of rolling a 1? What's the chances of rolling a 2? Well, we know that there are 6 numbers, right? You can either get 1, 2, 3, 4, 5, and 6. So if I roll a 1, that's a 1/6 chance. And if I roll a 2, and again, What's the chance of doing one or the other? I'd have to add them to get, there's a 2/6 chance, because it's always based on one event, a mutually exclusive event. So what's the one event I'm talking about? All of the alleles that are in the population. So hopefully that's clear enough. Now, what we do in Hardy-Weinberg, because there's always going to be different letters, we use P for the dominant. Okay, so that's my A there. And we use Q for the recessive. And we noticed that all the probabilities or frequencies, and we're going to use frequencies usually as a fraction, okay, plus all the frequencies of the recessive or broken gene, and you can think of that as a little b in my example, have to equal 1. So in my work here, in this worksheet, what do I have? They tell you that the Hardy-Weinberg states that frequencies of alleles or genotypes in a pot remain constant from generation to generation. Why? Because there is no evolution going on if those 5 conditions are met that we talked about. Frequencies represent the fraction of alleles in the population, and they're going to give you this, P equals 0.8, and they're going to say the dominant is 0.2. So they're giving you that in this case. So I'm giving you that Q is 0.2 and P is 0.8. What does that mean? Q means that 20% of the time, or 20% of all the chromosomes in the population are what? Recessive. Okay? And then this P equals 0.8 means that 80% of all the dominant genes, or the dominant alleles, or 80% of all of the chromosomes in the population are dominant. Notice 80 plus 20 equals 100. Or if I do this math here, P is equal to 0.8 plus 0.2, and it equals the 1, which is 100. And that's really a very important understanding. Now, when I'm dealing with this, okay, when I'm dealing with this right here, we're dealing with total number of the frequencies in the population. We're not talking about individuals, we're talking about all of them. If we scoop back up to 3 individuals, and let's pretend we have 3 individuals in my small population, which wouldn't be a good thing for Hardy-Weinberg postulate, but if you look at the 3 individuals I have here, heterozygous dominant, homozygous recessive, homozygous dominant, sorry, homozygous recessive, and heterozygous, look at those 3 people. What would be the total percentage of alleles? Well, you'd have 3 out of what? 1, 2, 3, 4, 5, 6. So 3 out of the 6, which is equal to 0.5, would equal your what? Would equal your Q, which is your recessive allele. Let's get this out of the way. And what else would we have? Well, and just in my example, we would have what? 1, 2, 3. 3 out of the total 6 chromosomes would be dominant. 0.5 would equal what? Would equal my P. And P represents in the Hardy-Weinberg formulas the dominant gene. So that's how they're doing it. We're doing what? Total number of chromosomes, not individuals. We're not saying that 3/6 of the total individuals is a recessive. That doesn't make any sense. We're looking at total. Okay, and if you notice, if you add 0.5 to 0.5, you're going to get that 1. And that's what we're talking about here. Okay? All right. Now, if you're talking about getting the individuals, okay, it's different. Individuals are different than a total frequency, and this is really important. Yes, we have a P, which is the frequency of the dominant allele, okay, which in my case, I'm making what? A. I know I changed it from B up there. And then in this case, it's 0.8. So 0.8 or 80% of the population has a chromosome with the dominant A gene. And then we would say, well, Q, the recessive gene, exists 20% of the time, or 0.2. And of course, that represents the little b in my example. Okay, now that does not represent individuals. How do we get individuals? Okay, well, we need to use the multiplication rule to do this. Okay, now we care a lot about the individuals that are BB because to have the PKU illness, you have to have two chromosomes. Just because we know 20% of the entire population has the recessive gene, that doesn't mean it's 20% of the people have it. That just means 20% of the total number of chromosomes. So we want to move from the number of total chromosomes to individuals. And that is so important. If you don't understand this right here, you miss what Hardy-Weinberg's about. So let me move on to the next page and I'll show you what I mean. So I'm on to the next page and I'm using large A for my dominant allele and little b for my recessive. Okay, so in this set of problems, I already told you that I'm using 0.8 or 80% for the A and 20% for the B. That's something that we're starting with just to understand how this all works. Now let's do a cross between two heterozygotes to see how this all works. Now this cross of the two heterozygotes works because of the way of the total choices occur. And let me explain. If you think about it, what are the two possibilities of the alleles? They're A plus B, right? That's essentially what we have in the population. So that's the total amount of alleles. And if you're going to cross them with the same population, now we can't add this to something else. We're going to multiply this, and we'd multiply this by, guess what? Again, this is the total amount of alleles in the population, A plus B. Okay, and actually this would be the frequency when we add them together. Wouldn't this really be 1? In this case, it'd be 0.80 plus 0.2, and it'd be 1. Okay, so if I cross this with someone else in the population, wouldn't the combinations also be A plus B? Of course, those are the total. And that would be times the same thing, 0.8 plus 0.2. But this time I'm multiplying because when you cross and make zygotes, okay, it's an individual or a separate roll. This is the total amount of choices in one roll. This is the total number of choices in another roll of a dice, or at least a coin if you want to think of it as two sides, A or little b. And when you make zygotes, brand new individuals, okay, now we multiply because these are independent events. What's independent? Oh, don't lose sight of the chromosomal line of inheritance. The independent event is which of these alleles are we going to use? Is it going to be two little b's that come together to make the zygote? Two a's? We can't always assume that the individuals are heterozygous, but when we consider the entire population and the frequency of those alleles or genes on a chromosome, we can make that understanding of an AB crossing with an AB. Those are the total choices, and we multiply because, again, making one zygote is different from making another. They're separate roles. So we're going to cross all the possible A's with all the possible B's, and we're going to see what this will look like in terms of individuals. We're starting with the percentages of the alleles in the population. So I can multiply the total choices in the population with itself, okay? Because they're making zygotes. I'm pulling together one chromosome from another, and we should be able to figure out what the frequency of the three different types of individuals. And what are they? They're going to be homozygous dominant, heterozygous, or homozygous recessive. We're going to find the frequency of those individuals. This number is the frequency of the total amount. In this case, A represents the dominant. So let's do that cross. Obviously, what we have here is we're going to have two A's. And we're going to have an A here and an A here. And then we're going to bring in, okay, the B comes across, okay, B comes down, and then we have eventually, as we suspect, okay, a homozygous recessive gene. Now, what does this mean? Well, when you take the frequency of the P and times the frequency of another P, what do you think the new frequency is? And if you haven't figured that out, it's going to be 0.80 squared, which is equal to p squared. P squared represents the total individuals. What does P represent? P represents homozygous A. So when you have A and A zygotes, that's P squared. If I can guess here, now you have A and B. Well, you have A, which is 0.80, times what? Times 0.20. What's the 0.20? Okay, that's the little b. And that's equal to PQ. P times Q. Same thing over here. 0.8, this is the same exact thing. So it's 0.8 times 0.2, it's equal to PQ. And these are individuals. Individuals have what? Pairs. And what about K, in this case, BB? Okay, the homozygous recessive. Well, that's going to be 0.20 squared, which equals Q squared. Q represents what? The recessive allele. So you need to understand, party people, that when we start talking about individuals, you have to have two different alleles there. So these are the individuals. And we can calculate what the frequency of these individuals are by using this formula. Now, real quickly, since you guys have probably seen quadratic formulas, P plus Q from one adult to another adult gives you what? Well, I learned to do the quadratic by the method of FOIL. You guys may have learned something else. So I do first, so I'm going to do P and P, that gives me P squared. And I'm going to do outer, P times PQ, so that's going to be plus PQ. And then we have inner, I, so this was the what? The outers. And then we'll go inners, which is these guys right here, that's the I. So it's going to be PQ again. And then we have L for last, and that'll be Q and Q. And all I'm doing, for those that don't understand, I'm basically solving for an aquatic equation, or at least I'm factoring it. So in any case, we would go plus Q squared. And all of this has to equal what? Because you know math, you'd probably say 1, but you should know better the frequency of this individual, homozygous dominant, plus the frequency of what? The heterozygotes, plus the frequency of the recessive, okay, homozygous recessive individuals have to equal the total number of people in the population. Now we can simplify this. Of course, we can add these together. P squared plus 2PQ plus Q squared equals 1. So to frequency of genotypes of individuals, they're going to have a pair. In this case, it's going to be P squared, okay, which we know is 0.8 squared. Put that in your calculator, you get 0.64. What does that mean? 64% of all the people in the population are what? Are homozygous dominant. What about K? What about little a, big A, little b? The homozygous little a, little big A, little b. I can do this. The homozygous recessive, homozygotes, I should say. I can do this. Well, it's right here. So it's 2PQ. So 2 times 0.8 times 0.2, and you put that in your calculator, and you get 0.32. And what about the homozygous recessives? Well, that's just Q squared. These are the individuals. And that's equal to 0.2 squared. If you need a calculator, go ahead. If not, 0.04. Okay. And if you haven't already noticed, if you were to add all of these numbers together, you should see that we get the 1, because what are these? These are the probabilities of all the choices in one roll, if you think about it. And you can add them, and they'll equal 1. Okay? So understand, very important, this represents individuals' frequencies, whereas the individual, okay, this right here represents what? the frequency of just the what? Chromosome that has that gene in the population. It's important. We moved from the frequency of the total chromosomes that have that gene, and we moved toward the frequency of the individuals. Look at this, 0.04% of the entire population will be what? Homozygous recessive. That means 0.04% of the population will have PKU. Oh, we brought that back into biology, assuming what? There is no what? Evolution going on. This is my baseline. Okay. So how do we use this? We can now, and before we use this, let's take a little tour of our reference tables where we're now going to see where they show us where individuals versus total alleles are. So let's go to the biology, AP biology reference tables. And bam, there it is. Okay, so here's our AP biology reference table that we've spent a lot of time on using some of the statistics. And let's scroll down. Laws of probability, we talked about the multiplication rule, even in Hardy-Weinberg, and the addition rule, the mutually exclusive rule. And there we go, party people, Hardy-Weinberg equations. Notice p squared is 2 what? Dominant alleles. That's individuals. So the frequency of the dominant allele in the population is p, and this is the frequency of the recessive is q. They tell you that. And you need to understand that these are individuals. These are the individuals that are what? Homozygous dominant, heterozygotes, carriers, so to speak, like they call them. And then, of course, people, if it is a recessive type of disorder, people that are inflicted and they have the homozygous recessiveness, okay? And this, again, is used for the total population. These are not individuals. You can't have one allele. You always have a pair. So this is the total amount or frequencies that has to add up to 1. These are the frequencies of total chromosomes with those genes. This is the frequency of the total people in the population. Notice they have to equal 1. That's an important distinction that makes this so much easier to understand. Let's go back to the last piece of this worksheet. Okay, so let's start looking at, let's say, a typical problem using Hardy-Weinberg. Now, I know that I started you with the 0.8 and 0.2, but now we're going to do this problem another way. So we're going to use real data that could be given to you in a separate problem. So all the numbers that we used don't relate to this problem. However, all the concepts do. So they say the occurrence of PKU, as we talked about the homozygous recessive, and they have 2 little k's to represent what they want to talk about. I was using little b's, it does not matter, is 1 per 10,000 births. So 1 divided by 10,000, meaning 10,000 is the total number of what? People in the population, and there's 1 in them. That's the percentage. Part over total, okay, will give you a fraction. If you times it by 100, that gives you a percentage. But we're going to use that as a frequency. So a frequency is a part over the total. If you do part over total population times 100, that will give you a percentage. And normally your frequencies are the fraction like this is. So the part is 1 over the total 10,000 people, and that gives me that fraction. And that's the frequency of people that have PKU and let's say our population. Okay, so the frequency of individuals that are afflicted, now I understand, is what? Now, this is the number of people who have the disease. We know it's a homozygous recessive. So this is really important to realize. By me giving you in this problem the people who have the disease, what I'm really giving you is I'm giving you the frequency of the Q squared. Remember, these are people. Q squared. Q is the recessive. If you forget, go to your reference table. Q times Q, that means you have 2 recessive alleles, is the same number. Okay, that's really important that you see that right away, that number for the frequency of the individuals that have the what? The disorder is the same as the frequency that's Q squared. These are people, okay, Q squared. Now, to find the Q, which now a lot of times they want to know the frequency, they want you to go back because they want you to go back often to see how many of the others. To find the Q, all right, first, well, let's say I want to find the P. What does P equal? The people or the frequency of the what? Dominant gene. In order to go backwards, I know that P plus Q equals 1. But right now I don't have Q, I have Q squared. So to find the Q, or the frequency of just the Q in the population, all we do is take the square root. So the square root of Q squared, which is the individuals that have the disorder, is equal to the what? Okay, the Q. So the square root of Q squared gives me the frequency of the total number of what? Chromosomes in the population that are recessive. So let's do that. So take the square root of 0.0001. And when you put that in your calculator, okay, what you're going to get is 0.01. What did we just solve for? What did we just solve for? We solved for Q. We just found the frequency of the people in our population, or the frequency, sorry, of the number of chromosomes that have the recessive allele. It's 0.01. Can I find P now? If I know Q is 0.01, and 0.01 plus what number is equal to 1? All right, well, minus 0.01, and you're going to get what? 0.99. So from this math, okay, P, which is equal to the frequency of the dominant chromosomes, okay, is equal to 0.99. It's not P squared, it's not individuals. Okay, so we just found that P is equal to 0.9. That 99% of the total alleles, total chromosomes in the population have the dominant allele. Okay, so what's the frequency? So I found that to be 0.99. That's one answer. We found the frequency of the Q allele to be 0.01. That's the other answer. Now what's about the carriers, the homozygous recessive? We do 2 times the P. What's the P now? The P is 0.99 times the what? 2PQ. Now where'd that come from? Up here. Right? Remember this? 2PQ? Because there's 2 of them in these crosses. 2 of them. 2PQ. That's where it comes from. Don't lose sight. But it's given to you in the reference table anyway. So 2PQ. So 2P, what's the Q? 0.01. Oops. 0.01. Put that in your calculator, and you get 0.0198. And that's the frequency of the what? PQ, or the what? The heterozygous individuals. All right, this is the individuals. And this is approximately what percent of the population? Okay, what percentage of the population is heterozygous? Well, what do I do here? Well, 0.0198 is essentially, if it's a frequency, well, to get it back out, 0.0198 is a frequency. Remember, we times our frequencies by 100 to get our percentages, and we move this over 1, 2, and it becomes approximately 1.98% of the population is what? Is heterozygotes. So let me just review this problem. In this problem, I gave you the number, or the probability, or the frequency of the people that were what? Homozygous recessive. I gave you that. You had to know that meant that that value was Q squared. To answer what part of the population, okay, was heterozygous, we had to find our P and Q. So if I know this is my Q squared, these are individuals, hello, squared, both of the recessive, I did what? I square rooted my Q squared to get Q. Once I had my Q, I know that this is always true. I subtracted that from 1 to get my what? my P, which is the what? Dominant genes. Okay. And then, of course, once I have my P and my Q, I plug it into my formula 2PQ, and I got my frequency of individuals. Okay. And then I convert it to a percentage by moving the decimal place over because I'm timesing by 100. Don't forget, a frequency is part over total. A percentage is part of a total, times 100. All right. All right, so let's make up another type of problem. Okay, let's start with a great little example of what a Hardy-Weinberg problem will look like. Okay, so on the back side of this, or a separate piece of paper, I want you to keep following along so you get a really good feel for how this works. Okay, so what we have here in this great example here, at least I hope it will be, okay, we have black moths that consist of 13 individuals that have the black allele. And I have to tell you in this that this represents, let's make it two little bees. Let's say they have the black color, or the black color on the wing of this type of moth is homozygous recessive. Okay. Now, to have the white colored moth, or the white body on the moth, all right, it's due to the dominant gene, so it could be homozygous dominant, but it also could be a carrier for the black, right? Heterozygous, a little b. So there's a problem here because when we deal with the white moths, they can be two different genotypes, even though they have the same phenotype. And this is a very typical Hardy-Weinberg problem. And let me show you how this would be asked. So in this problem, knowing the total number of individuals that have this phenotype, they're going to ask you, hey, what is the frequency? What is the frequency of the homozygous dominant? What's the frequency of the heterozygous, or the carrier? Both would be white, but we know they have different genotypes. So what we do is we're going to add these total individuals up. And when we do so, we get 613. Now, what I'm going to do, I can't really start from the white moth because I don't know if this white is either homozygous dominant or heterozygous. So I'm going to start from my recessive allele. That is absolutely vital. So we're going to start right here. This number of moths I know definitely, okay, is little b, little b. Okay, individuals, because these are moths, so I know that for sure. That's the only starting place I have. I can't start here because, gosh darn it, I don't know which one it is. If it's white, it could be white for two different genotypes. Important. So what I'm going to do is put 13 over 613. What does this give me? As I said before, this is the part over the total. Total number of what? Individuals. And so when I go 13 divided by 16 in my calculator, okay, I get 0.0212. That's the frequency of the individuals that have the black body, which we know is due to their recessive, homozygous recessive. So my friends in chemistry, or biology, I should say, it's equal to Q squared. We found the individuals. Why Q squared? Q is for the recessive, and these are two. Remember, to be black, you have to have two recessive alleles. So this is my Q squared. Now think with me for a second. I'm trying to find what? The frequency of this guy and this guy. All I need to know is what? The frequency of my recessive allele and the frequency of my dominant allele, and I can do this. But right now, I don't have the frequency of the allele. I have the frequency of the individuals. So what I'm going to do is I'm going to take the square root of my Q. Square root of my Q squared is my individuals, will give me my Q. I'm going to go from individuals to the frequency of individuals to the frequency of the alleles. So I do that. So watch me do it. So I take the square root of 0.0212. And what does that give me? Okay, that gives me a Q to equal 0.1456, and that equals my Q. Now, what does that mean? That means that the frequency of the entire population of chromosomes, 0.1456 of that total population of chromosomes, okay, have the recessive chromosome or allele. Okay, very important. Now I can work with that. Okay, because again, what am I after? Like most Hardy-Weinberg problems, I'm after homozygous dominant, which we should all know is what? The individuals that are homozygous dominant are what? Not Q squared, P squared, right? And the individuals that are recessive, well, isn't that 2PQ, right? So I need the individual P and Q alleles, not the individuals, right? I need P squared would give me that. So I need the value for P, all right? And I need the value for P to square it to get my AA, all right? Or I need the value of what? P to get my homozygous. So watch what I do here, party people. I have my Q, and what do we know? Once I have my Q, I can get my P. Why? Because P plus Q equals 1. What does this mean? The frequency of the dominant plus the frequency of the recessive allele must equal 1 in my population. So if I know the Q is 0.1456, P plus 1 number equals 1, do your algebra, and when you do that, you're subtracting 0.1456 from 1, okay, and what you get is P, the frequency of the dominant allele, is 0.8544. That makes sense. If I know the frequency of my recessive allele in the entire population, and there's only two what? Chromosomes and 2 different alleles, okay, then the other one is 0.855. Must make sense. So that's another piece of information I need. And now I can do this. I found the individual frequencies for the alleles in the entire population. And now I'm going to use them to find my individuals. When you have your individuals, what's the formula? P squared, homozygous dominant, plus 2PQ, which is my recessives, my carriers, which is half recessive, half dominant, right? The heterozygotes, plus my Q squared, my recessive, homozygous recessives. So I was after in this problem, what is the frequency of the individuals that are dominant? In this case, it's AA for the white moth. And who is the heterozygous? A little b. So I'm after this. Well, I have my individual numbers. What's P? So to get P squared, which is the individuals, I square this number. So 0.8544, and I square it. And when I do so, I round a little bit and I get 0.730. Well, what's that? That's the frequency of the individuals that are homozygous dominant. What are the frequency of the individuals that are heterozygotes, so carriers? Well, how do I do that? 2PQ. So I do 2 times the individual of the P, the frequency of the P in the population, which is 0.8544, times the Q, which is 0.1456. And when I put that into the calculator, all right, I get 0.249. Okay, so those are my answers. Okay, so my answers to my question is the frequency of individuals that are P squared, which would mean homozygous dominant is this. The frequency individuals that are heterozygotes, the carriers, is this. Now, really fun, what was the percentage of the people that were recessive? We found that to be what? Q was equal to, well, Q squared is 0.0212, right? So doesn't it make sense that 0.73 plus 2.49 plus what was this value that we got? 0.02 approximately, 0.0214, I believe. 12, my short-term memory's gone. So 0.0212 was our q squared. If you add these up, this plus this plus this, you would get what? 0.999. So, Mr. Grotsky, isn't it supposed to equal 1? It does. And because of our rounding, okay, we didn't get that. All right, remember, this is the formula for the individuals. All right, where was our starting point? Hint, hint, hint. Where is our starting point? Always the homozygous recessive, because that number of individuals will be what? Will be your q squared. 13 over the total here, that'll be your q squared. And you can't do that with the white moths because they have two what? Different possible genotypes. So therefore, you can't do that. So this is typical of given an amount of each, two different, let's say, phenotypes, and figuring out what the percentage, assuming there's no equilibrium. Now you may Mr. Grotsky say, well, okay, how do we use this when there is I say equilibrium, no, I should say no evolution, based on the 5 parameters. Why would we use this? How could we use this to see if there is evolution? Well, once I determine my frequencies of my what? Of my individuals, let's write them here, 0.730, 0.249. Okay, let's get rid of these guys. So once I have the frequency of my individuals, I can check to see over some time period if these frequencies change. So if these frequencies change, there must be some selective pressure. Now you understand how we use this as microevolution. So if I know this is my baseline with no evolution, and there appears to be a selective pressure, I can calculate what the frequencies of these alleles are using the same mathematics. And if they're changing, then I know there is some selective pressure and there is evolution. Even though we're not going to see a new type of moth appear with a new morphology, we can see changes in the what? Allele frequencies as our what? Evidence of evolution. That's why we call this microevolution. Or at least we can look at if in fact the allele frequencies are changing and causing an evolutionary change before we see huge types of macro changes that we've been learning in class. Hope this helped.